Hey guys, this video is about some early microbiologists. One of the first ones we're going to look at here is Robert Hooke. And he was actually famous for looking at cork cells. And when he looked at cork cells, he actually termed our word cells because he looked at an image much like this one right here, and he looked at these little units and he said, gosh, that looks like these cells that I see in monasteries. And so he's the first one to coin the term cells, and he does so because he's using a very, at his time, revolutionary tool called a microscope, which allows him to magnify the living world and see things that are much smaller. Another scientist that we talk about is Anton Van Leeuwenhoek. And Leeuwenhoek observed, again through a microscope, and he observed protozoa in pond water, and he would see protozoa swim around. And so he called these protozoa like little mini animals because they would swim around and they seemed to be hunting or gathering food, kind of like you'd expect an animal to do in the wild. These are two of our more notable scientists that worked early on in the field of microbiology. But of course, they had to wait for the microscope. Without the microscope, we weren't able to see this world, and so this unseen world became available to them because of the microscope. These are some of the earliest drawings of what Anton Leeuwenhoek saw. So if you look here, he's got this particular one right here, which probably would have been a very simple protist, and it's got a little dotted line pathway here, and that's kind of what he saw it moving in different directions because it's kind of hunting, and so he saw a lot of different sized things in that pond water, and to them this would have been a brave new world. Another important scientist was Lazzaro Spalzanetti, and he lived from 1729 to about 1799, and he worked to debunk the idea of spontaneous generation. And this idea is really one that prevails for a very long time, even after there's some very significant evidence against it. And it's the idea that living life or living matter or living organisms will just pop up out of nothing. So you'll take non-living and it will become living. And he's going to work pretty heavily to debunk this with some of the work of Francisco Redi, this idea of boiling soup. And so what you take is you take a flask and you put broth in it. And when you boil it, when you actually boil this flask, it's going to kill the microorganisms in it. That's typically why if your water source isn't clean, they tell you to boil it. So you kill the microbes, and then it's sterile. There's nothing in there. You seal it up so that nothing can get in. You can leave that for a very long period of time. Nothing will grow because you've effectively killed all microbes in there. But once you break that seal, at that point in time, you start to have microbes grow again. And so what this shows is evidence against spontaneous generation. It says, look, if it's coming out of nothing, why didn't it come out of nothing when it was sealed? If it needs nothing to become alive, why didn't it do it then? It only did it when we broke the seal, and because of that, spontaneous generation can't possibly be true, or else it would have happened here. We also have him observing cell division. So he's one of the first scientists to observe cell division. Discovering that cells were the basic unit of life is part of our cell theory, and we have two scientists kind of to thank for that. First one is Matthias Sleeden, and he's going to conclude that all plants are made of cells in 1838, and he does this because he's going to look at a bunch of plant cells like these right here. And he's going to look at plant cells of hundreds and hundreds and hundreds of plants, as many as he can possibly get his hands on. All of those, again, under the microscope. And every single time he's going to see a cell. And if you look here, these are the cells we're talking about. Now, if you know a little bit about plants, these little green dots in here, you should know what they are. Those are chloroplast structures, and they're actually going to allow them to photosynthesize. And so those green structures are part of that cell. Well, Matthias Sleeden is going to look at as many plants as he can, and when he cannot find one that is not made of cells, he's going to conclude that all plants are made of cells. Theodore Swan is going to do something very similar in 1839, so not much later. He's going to look at as many animal cells as he can possibly look at, and when he fails to find one that is not made of cells, he's going to conclude that all animal cells are made of cells, and all animals are. That's their basic unit of life. And that's important because it really sets us up to explore the cell as a fundamental unit of living things. Heinrich Anton de Berry is another important microbiologist. In 1831, he was born, lived through until 1888. He observed conjugation of algae, which is actually how they exchange genetic material. And that's pretty important because it allows them to be very dynamic and deal with some situations because they have a good genetic diversity. He's the first one to describe cellular reproduction of fungus, or the mushrooms you might be used to seeing. You might think that the mushroom structure you see on the ground is all of the mushroom, but really the mushroom is all throughout the ground, and that's just its fruiting body. But one of the more important things that Heinrich would have done was he studied that fungus that get infected a plant. This is pretty interesting because at that point in time, he's going to set the basis for the fact that diseases are caused by some organisms. He also studied lichens, which are a symbiotic relationship between a fungus and an alga. And that's pretty important because the first term symbiosis is coined by him, which we now define as a close permanent interaction between one or more organisms. Louis Pasteur is very, I think, well known, at least for the pasteurization process. You might think that the milk you buy at the store is perfectly sterile, but it is not. It is pasteurized, and similar to what Louis Pasteur would have done. He worked early on with yeast, and he showed how yeast can break down sugar. And you might think, well, okay, microorganisms eat, big deal. But in his time frame, when he was studying this, that was important. And he continued working with this yeast in the wine industry. So he worked in the wine industry, and what he found was that he was going to be able to solve some bitter wine problems. He did an experiment with this S-shaped flask where he's going to, again, boil something, and this S-shaped flask does something pretty special in that, microorganisms, they're all throughout this, right? All this area is full of microorganisms, but they can't get into that S-shaped flask. You'll notice that right here that they're blocked. And so he can leave that. He can leave it over and over again. No big deal. It won't grow. As soon as he turns that S-shaped flask, and more importantly, as soon as he exposes it to this part of the flask that has bacteria in it, at that point in time it then grows. Now, you might think back and go, hold on, wasn't that just like the boiling broth experiment? And it was, but spontaneous generation is still around in his day, and he's got to continue to debunk it. Again, pasteurization is something he's known for. It lowers the amount of microorganisms in our milk or other things so that it's at a reasonable level. That's why your milk expires. The bacteria grow too much. These have been just a few of many microbiologists, highlighting some of the things that they've done. Hopefully this has given you some appreciation for the world around us and shown you a couple experiments that were enabling them to debunk, for instance, the spontaneous generation theory, that cells came out of nothing and that life was able to be created out of non-living. And it showed you, I hope, that from their perspective, it was a brave new world where they're studying something nobody had ever studied before.